Here we are, Unit 3, Cellular Energetics, and this is video 4. In this video, we are going to be focusing on photosynthesis and a bit of fitness as well. So Units 3.5 and 3.7 of AP Biology. So let's first start with the why and the how of photosynthesis. So the why question, the ultimate question, is what is the fitness advantage to organisms that can photosynthesize? And then the how, the proximate question, is that there are two stages of photosynthesis. What are those two major stages and where do they occur? So the reason why organisms photosynthesize is that capturing light energy and transforming it into chemical energy allows the organisms to power reactions of their cells, which allows organisms to survive and reproduce. So it's taking energy that's available from the sun and turning it into a usable chemical form. And the two phases of photosynthesis are light dependent and light independent. And now we're going to go into a little bit more detail on both of those. So where do these two phases occur, light dependent and light independent? And what are the reactants and products of each phase, as you can see in the diagram? And what are the steps within each phase? So the light-dependent reactions, the ones that rely on light energy, happen in the thylakoid membrane. And you can see that pictured on the left side of this diagram on the sort of green stacked circles. And there are three steps of the light-dependent reactions: photolysis, ETC, and chemiosmosis. In the light-independent reactions, which can happen in the light or in the dark, they don't require light. This happens in the stroma of the chloroplast, and this is known as the Calvin cycle. In terms of reactants and products, you can see those in the diagram. So you can see light, water, and carbon dioxide coming in. And you can see oxygen and sugar coming out. So those are the major reactants and products. There are also intermediates. So ATP and NADPH are intermediates that are produced by the light dependent and used in the light independent. And then they are sort of, the counterparts are regenerated as NADP plus and ADP. So we'll get into more detail about all of those as we go through photosynthesis in more detail. And these are just showing where those reactions are occurring. This is showing where the thylakoid membrane and the stroma are. So let's go into the first step of photolysis. Photolysis means water splitting, but look at the root word in there. So what is the root word of photo and what is the root word of lysis? Where does that term come from? So photo means light and lysis means break, but it's not light that's breaking, it's water that is breaking from the energy of light. So sunlight provides energy, which lyses or breaks water. So what is this going to produce if water breaks? What's produced are electrons, hydrogen ions, and oxygen. So I'm less concerned about the counts here. Don't worry about how many of each of these are produced, but just understand what is produced. So we've got electrons, we've got hydrogen ions, and we've got oxygen. And this is a reaction that requires a lot of energy to happen. And the energy comes from the sunlight. Look at the oxygen on the left side and look at the oxygen on the right side of this diagram. What happened to oxygen? And in this case, oxygen was oxidized. This is actually fairly unusual. Usually oxygen causes the oxidation of other molecules, other atoms. But in this case, oxygen was actually oxidized. So oxygen lost control of electrons. Before we go into ETC, I want to take a look at a molecule called chlorophyll, which you've probably heard of. Please don't be alarmed by that diagram on the right. You certainly don't need to know the structure of chlorophyll, but I just wanted to be able to see it and see the different parts of it. But again, you don't need to memorize that. You don't need to know it. But what is chlorophyll in general? What's its function and where is it located? So chlorophyll is a light-absorbing pigment molecule, and it's located in photosystems. Here you can see a picture of where this is. So photosystems are embedded in the thylakoid membrane. So you can see from the diagram a chloroplast, and within the chloroplast is a stack of thylakoids. And then within the membrane of the thylakoid is a cluster of chlorophyll molecules. And then you can see a sort of zoomed-in look at the molecular structure of chlorophyll. You certainly don't need to memorize that. All right, so a little bit more context. Photosystems is that area where light energy is absorbed, and those are part of the thylakoid membrane. This is where photolysis takes place on the inner side of the thylakoid membrane. And the chlorophyll's role is that it allows the light energy from the sun, or from a light, to excite electrons. And then that energy is going to be transferred along the way in the electron transport chain. And we will see that in a moment. So here is the electron transport chain. There's a lot going on in this diagram, but what I want you to focus on is what's happening to the electrons, and then also what do the blue and red squiggly lines represent. So the basics of what's happening to the electrons is that they're gaining energy. That energy that they're gaining comes from the sun, and it's those chlorophyll molecules that are facilitating that reaction. So what you can see is in this diagram is that the energy of those electrons gets boosted up from the light energy, and then those electrons get transferred during redox reactions as part of the ETC, and their energy goes down. And then they get boosted up again by another photosystem, and then as they go through the electron transport chain, their energy level goes down again. So what the blue and red squiggly lines represent is the light itself. Photosystems and chlorophyll don't absorb every wavelength of light. We'll get to this more at the end of this presentation. But so that blue and red squiggly lines are representing the light that they do absorb. So let's look at a few more details of the electron transport chain. First up, let's make sure we have a good definition of what ETC is. So stop now and define ETC. ETC stands for the electron transport chain, and it's the transfer of high-energy electrons from one molecule to another through a series of redox reactions. So we have one molecule reducing and one molecule oxidizing. The electrons lose energy along the way, but a hydrogen ion gradient is produced. And you can see that happening in step 2 of this diagram, where you can see the path of the electrons in red passing from one molecule to another. And along the way, the hydrogen ions get shuttled through from the outside of the thylakoid into the inside of the thylakoid. And you can see that hydrogen ion gradient building up, representing, because there are higher numbers of, or higher density, higher concentration of hydrogen ions on the inside compared to on the outside of the thylakoid. Next question I have for you about the ETC is, what is the terminal electron acceptor of ETC? And to help you answer that, think about what terminal means and what acceptor means. The terminal electron acceptor of the ETC in photosynthesis is NADP+, because that's the molecule, the final molecule in the electron transport chain that picks up that electron. And you can see that in the kind of orange diagram, or the orange arrow shown on the right side of this diagram. So NADP+ combines with a hydrogen ion and 2 electrons to create the electron carrier NADPH. Again, I'm not concerned about numbers here in terms of how many electrons, how many hydrogen ions, but I do want you to understand the difference between an electron acceptor and an electron carrier. The electron acceptor is the molecule before it has the electron, and the electron carrier is the molecule after it has the electron. So NADP+ is the acceptor, and then after it grabs that electron from the ETC, it becomes NADPH. This is now carrying that electron, and that's going to be important in a later step of photosynthesis. All right, so the next step within the light-dependent reactions of photosynthesis is chemiosmosis. Chemiosmosis is specifically the movement of hydrogen ions. So remember, we built up that hydrogen ion gradient in the ETC in step 2 of this diagram, and now in step 3, we are using that gradient. So chemiosmosis uses the energy stored in the hydrogen ion gradient across a membrane to drive cellular work, such as ATP synthesis. So if you can think of it like a river, if we put a dam in the river to build up a lot of water behind the dam, and then there are maybe a water wheel within the dam that spins as the water rushes by. And that can capture that gradient, that can capture the energy from that gradient to transfer it into a different kind of energy. So we are taking the energy of that gradient and using it to create ATP. One last term in this section, which is what is photophosphorylation. So break this down and let's look at some of the roots here. Photo again means light, and phosphorylation means that we have a phosphate group added to something. What are we adding a phosphate group to? Well, we're adding a phosphate group to ADP to make ATP. So photophosphorylation is all about making that ATP. Photophosphorylation is focusing on this process of both the ETC and chemiosmosis in combination to make the ATP in photosynthesis. So at the very basics, photophosphorylation is the combination of ETC and chemiosmosis in photosynthesis. The technical book definition I've also listed. It's the process of generating ATP through chemiosmosis using a gradient created by the ETC during the light reactions of photosynthesis. But really all that's saying is that it's the combination of ETC and chemiosmosis within photosynthesis. So we're making ATP, we're adding a phosphate group to ADP, we're making ATP using the gradient set up by the ETC. All right, so let's just do a summary. What are the two products of these steps? What do we get out of these steps of photophosphorylation? The two major products that I want you to be paying attention to are NADPH, which is our electron carrier, and ATP. All right, so maybe we should just stop here. We should congratulate ourselves. The organism made ATP. Why not just stop now? Why is there another step of photosynthesis? We made ATP. Why not just stop and celebrate? Remember that the major goal of photosynthesis is to convert energy into a form that can be stored and transported. ATP can't be very easily stored and transported. It's really good as a quick energy source, but it's not good for sort of long term, and it's not good for moving long distances around an organism. So we're going to use those products, the ATP as well as the NADPH, to create a molecule that is more easily stored and transported. What is that molecule? So the molecule that we get here is glucose. Glucose is much easier to store and transport than ATP is, and this is the ultimate goal of photosynthesis. All right, so here we are in the Calvin cycle. Let's understand what we're looking at here without getting overwhelmed by details. And to do that, the first thing I want you to answer is, what are the reactants of the Calvin cycle and where did they come from? So there are three major reactants. ATP and NADPH are made in the light-dependent reactions, and CO2 is another reactant that comes in from the environment. So plants and other photosynthetic organisms need to have a way of getting a supply of carbon dioxide from the environment in order to have the building blocks to make the glucose of the Calvin cycle. I recognize there are a lot of details in this diagram, but I want you to get practiced at looking at a complicated diagram and finding what's the most important. So the most important thing that I want you to focus on here are the reactants and the products. So as a reminder, we bring in carbon dioxide and we use the power of ATP as well as those high energy electrons that are being carried by NADPH, and that powers these reactions that turn carbon dioxide, which is just a single carbon molecule, into a 3-carbon sugar. So what kind of reaction is this if we're taking small molecules and we're building them up into bigger molecules? Would this be catabolic or would this be anabolic? So this is an anabolic reaction because we're building those molecules up. So let's go over the basics of what we just said, because there are a lot of complicated things and I want to make sure that you understand the basics. So first up, what is the overall goal of photosynthesis? Then there are 2 stages of photosynthesis. What are they? Where do they occur? And what are the 3 steps within the first stage? What's the importance of water, electrons, light, NADP+ and NADPH, as well as carbon dioxide? And you should also be able to diagram the process of photosynthesis and describe each of the 4 major steps. I'm not going to go over those right here in the video because that's what we just did. So feel free to rewind, go back to any part that you had confusion on, but these are some questions that you should be able to answer. If you're still having trouble with these concepts, there are lots of great videos out there. I always recommend Bozeman, I recommend Amoeba Sisters, Crash Course has great videos, and then here are a couple of others. Photosynthesis on the left, the title is Photosynthesis, Light Reaction, Calvin Cycle, and Electron Transport, which goes through the basics really clearly. And if you want to get a better idea of the Calvin cycle, there's a TED Ed video that gives you a pretty good idea of what's going on. Alright, so the last part of this video is going to be focusing on fitness. So first of all, what is the definition of fitness? This is more of an evolutionary concept, but it comes into play in every single unit of AP Bio. So what is fitness? Fitness is an organism's ability to survive and reproduce. So when we're talking about photosynthesis, let's think about what would enable an organism that photosynthesizes, what might help it have a higher fitness level. So the question I want you to think about here is, why does chlorophyll make leaves green? And related to that, what is the fitness advantage of having 2 types of chlorophyll? So take a look at the graph sort of on the bottom right, and I have 2 different chlorophyll molecules as reference here because they're mentioned in that graph. But focus on the graph. What's the fitness advantage of having 2 types of chlorophyll? The reason leaves are green is that that's the wavelength of light that is not absorbed by the photosynthetic organism. So if you look at the diagram, you can see that the wavelengths in the blue region and the sort of orange to red region are absorbed, and the wavelengths in the green region are not. And so it appears green to our eye because that's what's not absorbed by the leaf, and it's what bounces back and is able to be absorbed by our eyes instead. So the benefit of having 2 types of chlorophyll is that it can capture energy from a wider wavelength span of light. So if you look at chlorophyll A, the optimal wavelengths are different than chlorophyll B. And so organisms that have both of these can capture light over a greater range of wavelengths. Or if an organism is exposed to a certain wavelength of light, then it might favor one of these types of chlorophyll over another one. So in every unit, I want you to be thinking about how does each of these molecules, how does an organism's behavior, how does everything relate back to evolution, relate back to fitness and the idea of what's going to help this organism survive and reproduce. Okay, that's it for this video, and in the next video, we'll talk about cellular respiration.